In this video, or at least in the next two videos, I'm going to cover lambda particle morphology and also T4 lytic cycle and temporal gene expression. And our goals in both of these videos are to gain a more detailed understanding of the phage lytic cycle, to be able to apply our knowledge about promoters and gene structure to understanding temporal gene expression in the phage lytic cycle, and also to be able to apply our understanding of gene structure and phage biology to our analyses of our phage genomes that we're looking at in class. So the first thing that I think is important is that you're able to make predictions about the genomes that you're annotating. And I'm using the example of Sally's special genome because it's very small, and the majority of the genes in this genome are actually structural proteins. So can I use my knowledge about the phage genome synteny and the lytic cycle to predict gene function? So in the next two lectures, I want to make sure you understand the fact that phage genomes typically have certain genes that are more or less always in the same position and in the same order. And this is particularly true in the structural genes, which are highly conserved. And so in this genome here, I don't have the functions of these genes presented because I don't want to give that away to the folks that are annotating Sally Special. But because I know something about the order in which structural genes occur on most phage genomes, I can make some pretty good predictions about what the functions of these genes are. And that's going to really help me make decisions as I look at data for analyzing functions of genes. So can I also predict where promoters should be located on this genome? And we've already covered how to look for promoters in a genome and what a promoter looks for. But this is something that we're going to be asking you to apply in this unit as well. And finally, based on what I understand about the lytic cycle of phage and also how phage control gene expression in the lytic cycle, can I predict what type of promoter I might find in different regions of this genome by putting together what I know about promoters and what I know about the lytic cycle and the types of and how the genes in the lytic cycle are controlled. So caudovirales includes the tailed phage, and we've talked about these guys for the last few months, the Myoviridae, the Cyphoviridae, and the Podoviridae. And mostly we've been talking about Cyphoviridae, and that's mainly because the majority of tailed phage, about 96% of them, are made up of the Cyphoviridae. These are the phage with the longer, flexible, non-contractile tail and an icosahedron head. We've also talked a little bit about the Myoviridae. These have an icosahedron head, and they still have a pretty long tail, maybe not as long as those of Cyphoviridae, but they have a different shaped tail, and it's a contractile tail and functions a little bit differently during delivery of the DNA into the cell. And we're going to talk more about both Cyphoviridae today and talking about the structure of lambda, and we're also going to talk about Myoviridae today because we're going to learn about the lytic cycle of phage by studying the phage T4, which belongs to the Myoviridae family. And just as a reminder, although we don't talk about them much, there also are viruses out there with either short or no tail that belong to Podoviridae. So let's begin with the basic capsid structure of the Cyphoviridae phage, which includes the E. coli phage lambda. And the reason why I want you guys to understand this is because we're going to be looking at the genomes and starting to call functions for genes, such as the major capsid protein. And I want you guys to understand where we can find those particular proteins in the virion. So the major capsid protein is a protein that's, and the majority of the phage proteins in the capsid are repeated over and over again. And so the majority of the proteins in the capsid are often made up by one major subunit that's used over and over again. There are often other minor capsid proteins and proteins that decorate the capsid, but there's usually one protein that makes up the majority of the capsid head, and this is called the major capsid protein. There's also proteins that connect the head to the tail, and these are called head-to-tail connector proteins. And finally, the protein that forms the corner or the 12th vertex of the icosahedron. There should be 12 vertices in the icosahedral head. The 12th one that connects the phage head to the tail is called the portal protein, and in fact it's a very special protein because it forms the hole through which the DNA will exit the head and proceed through the tail when it's injected into the phage, and it's also the hole through which DNA is packaged into the head. And so you'll also be able to find this protein in your phage genomes. The major tail protein is the protein, it's a repeating subunit that actually polymerizes and forms the majority of the tail tube. And finally, there's a number of minor tail proteins that make up the tail fibers and the tip of the phage tail. So one thing that's important to understand and that makes viruses very different than cells is clearly they don't divide like cells do. They don't undergo fission or mitosis. The way that we end up with phage progeny is by assembly of a whole bunch of gene products, pooled gene products. And essentially the tail fibers are assembled, the heads are assembled, and the tails are assembled, and then the head and the tail are assembled together, and finally the tail fibers are added on. So there's 3 pathways that converge together to produce the final virion. And the particles are assembled from multiple copies of identical components. This makes it very efficient, and it actually contributes to the stability of the particle too, having like proteins binding like proteins. And finally, I want to emphasize that assembly is highly efficient due to the high concentration of structural proteins. Having that high concentration of pooled protein subunits drives the assembly reactions forward, making it more efficient. So the packaging of the DNA is very interesting, and in this lecture I'm only going to talk about packaging of DNA genomes that have COS ends, and Keith has already produced a video describing packaging of circularly permuted genomes, which is quite different. In any case, the DNA is packaged into the head to liquid crystalline density, which is very energetically unfavorable. So the DNA packaging process is actually an ATP-dependent process. And the DNA is packaged through the portal protein, which is this dodecameric complex here, into the head by the terminase in an ATP-dependent manner. In fact, the terminase is like a motor, a molecular motor. So let's look at that a little bit more closely. And again, I'm pointing out proteins that you will very likely see in your phage genome annotations. So the first thing that's assembled is a head or procapsid. And essentially what we have here is a number of chaperone proteins and scaffolding proteins that are used to build the head. And these scaffolding proteins and chaperone proteins are only there in the immature capsid head and are there to hold the structure and stabilize the head until the DNA is added. And then there's actually a protein called the capsid maturation protease, which will degrade these proteins, and they'll leave the head when the DNA is being packaged inside the head. And then we have a mature head with the DNA packaged inside it. So you should look for scaffold proteins and capsid maturation protease in your annotations. Now, the protein that packages the DNA is called the terminase. Sometimes there's one gene that encodes the entire terminase, but more often what you'll see is a small subunit terminase and a large subunit terminase. And these proteins work together to first recognize a concatemer of DNA. And so this is a long string of viral DNA units that were produced during DNA replication. So there's many, many, many genomes in one long string of the concatemer. It recognizes this double-stranded DNA and binds to it. And the terminase also binds to the portal proteins. So it's bringing the DNA to the portal proteins, and then it acts as an ATP motor, stuffing the DNA into the capsid head. And it will do that until it finds a COS end. It has endonuclease activity and will cut the DNA at the COS end, producing that sticky end, so that linear DNA is packaged into the capsid head with two sticky ends on the end, produced by the terminase. So this is kind of a neat protein because it has so many different activities. And you may notice if you find this protein that there are numerous types of conserved domains in this protein because it has different enzyme activities. It has helicase activity, it has endonuclease activity to cleave the COS ends, and then it has this ATP-dependent motor which translocates the double-stranded DNA into the capsid. So tail assembly in lambda is interesting too. So that really, really long gene in the genome, and usually this is the longest gene in the genome, although there are exceptions such as Sally Special, is the tape measure protein. And it's the tape measure protein because it determines the length of the tail. And you can actually measure the length of the gene and it will correlate with the length of the protein. And you can look that up online to figure out whether or not you can predict the length of your tail and then measure the tail in your electron micrograph to see if your predictions are right. In any case, there's also a protein called the tail assembly chaperone proteins, which I'm going to talk about more in just a moment. And these tail assemble chaperone proteins interact with the major capsid protein and help it polymerize, starting at the very base of the tail, help it polymerize, and here's the little green major capsid proteins forming, up to the end of the major tail protein until it reaches to the very top. Here's another look at that in lambda. We start with the tip. The tip is made up of a number of minor tail proteins and tail fiber proteins. Here are the tail assembly chaperone proteins, which in lambda are called GPG and GPGT. And here it is interacting with the tape measure protein and the major tail protein, and they assemble until they get to the top of the tape measure protein, at which point the tail is mature and the head comes along that's already been packaged with DNA and is added on. Now, what's interesting about this set of proteins, GPG and GPGT, the tail assemble chaperone proteins, is that these proteins are basically translated from the same mRNA. And this is due to something that we learned about last semester, which is the program ribosomal frame shift, where we have a piece of RNA that encodes for GPG, and then the ribosome will translate the RNA, producing until it reaches the stop codon, and it'll produce GPG. And 96% of the time, it produces the short protein. But you guys know that sometimes the ribosome comes across the slippery sequence, and it backs up one base, changing the frame, and produces a different amino acid sequence, and it bypasses that stop codon. And now we have a protein that's longer. It's the same amino acid sequence as GPG up to the slippery sequence. And then there's a different amino acid sequence due to the change in the frame. And so this larger protein is made only 3.5% of the time. And so this program ribosomal frame shift allows the virus to make two different protein products that, lo and behold, interact at the C-terminus where the proteins are the same. And it also produces the perfect ratio of GPG and GPGT for the function of the tail-assembled chaperone proteins. So here's another thing that you can look in your genome annotations. Can you guys find the tail-assembly chaperone proteins? And in some cases, you're going to be able to annotate the slippery sequence and where this frame shift occurs. In some of the newer genomes, such as QUK, this may be harder to determine, but you will likely find these tail-assembled chaperone proteins. So what I'm asking you to do is to use synteny to determine functions in the phage genomes. This is the genome of Badger, which is, I think it's a cluster A4 genome, and it's a genome that I helped annotate, and that's why I chose it. And you can see that, okay, there's some strange things on the very left end. There's an H&H endonuclease here. There's something called the tail protein, which isn't very specific. There's something called the lysis cassette, and I'm going to talk more about this in part 2 of this lecture. But there's a lysin A, a holin, and a lysin B, and hopefully you have some idea what these genes might do. But I'm calling this a lysis cassette because I know these 3 genes will always be clustered together. They have related functions, and their location in the genome is always together. Then we had the terminase protein, and in this genome there's only one terminase gene, and most genomes you'll see both a small subunit and a large subunit. The terminase is almost always followed by a portal protein, and that's followed by a capsid maturation protease, a scaffolding protein, major capsid protein. So you can actually use Badger as a model for what the order of genes are likely going to be in your genome. There's also something called head-to-tail connectors, which we talked about earlier. And there's not just one of them, but there's usually at least 5 or 6 of them right in a row. So if you find one, predict that there's going to be 5 or 6 more to follow. And some of the HH-pred data will give you really good hits to the tail connector proteins, but the next one might not give you a good hit. But we can call this just the same because we know that there's usually 6 of these clustered together. The major tail subunit is almost always followed by the tail assembly chaperone proteins. Here you can see that it's annotated with the programmed ribosomal frame shift. This is followed by the tape measure, and usually the tape measure is followed by a cluster of minor tail proteins. And here's another situation where if you find one minor tail protein,